Hello, my name is Annetta Linwood, and I'm here to present my presentation, my advanced group presentation 3 on hemoglobin, um, the binding capacity of heme, the different pH levels in the lungs, and how carbon dioxide um, interacts with water. So let's begin. So hemoglobin is a component of red blood cells. It is a protein that carries oxygen from the lungs, from the lung tissues. It also contributes to the transport of carbon dioxide and hydrogen ion back to the lungs, and that's also known as an honorary enzyme. So let's talk about the structure. So hemoglobin structure is a quaternary structure. It is a allosteric protein, which means that the allosteric protein means that the shape changes when it binds to a particular molecule. It has four subunits, two alpha units, two beta units, or you can be called alpha beta dimer. So it has four heme subgroups, each contain an iron molecule. Also, if you have if you can see in the photo, you have alpha 1 usually goes with beta 1 and alpha 2 goes with beta 2, and that's a tetradimer. So if you look at the photos, you can actually see the heme group, the beta chains, the alpha chains, and then you see the iron, which is in the middle, which is the iron molecule right there in the middle. And this is the comes from the red blood cell. So let's continue. So, how oxygen binds to heme. So, heme group consists of an iron ion held in a heterocyclic ring known as a porphyrin. Porphyrin consists of four pyro rings, and these are the pyro rings. So, you have one, two, three, four. Those are the pyro rings. Pyro rings. So, the iron ion, which is the site of binding. So, you have the iron ion, which is the site of binding, coordinates with the four nitrogen ions. So at this point, the fifth coordination is occupied with the proximal histidine, but the sixth coordination is unoccupied and available for binding oxygen. Now at this point, the iron ion is outside of the porphyrin ring because an iron ion in this form is slightly too large to fit in the porphyrin ring. So the oxygen binds in the sixth coordinate of the iron ion. And when this arrangement happens, the electron within the iron so that the iron can be smaller and fit into the porphyrin ion. So this is in a rearrangement of the electrons so that that iron ion can actually fit into that porphyrin ring. So after the oxygen binds, the bound oxygen is stabilized by the form of hydrogen bond with the distal histidine. Now at this time, once one oxygen binds, then the other four are more likely to bind. Okay, so now we're talking about the structural changes after it's bound. Now when the iron moves into the plane of the porphyrin, the histidine residue moves with it. So as we explained when the oxygen bound to the in the previous slide. So the oxygen moved into the iron moved into the porphyrin, and now the histidine moved with it. So the histidine is part of the alpha helix, which also moves. The related movement of the histidine containing an alpha helix alters the interface between the alpha beta dimer, resulting in a substantial change to the quaternary structure. The response to this is the T and the R state transition. So now it's going from a tense to a more relaxed, which increases the oxygen affinity. Okay, so other structural changes. So you have the deoxyhemoglobin. So oxygen not bound to it, so it's in the tense state. It has less affinity for oxygen and the non-polar configuration. So this is a deoxygenated state. So it's more tense, has less affinity for oxygen, and it's a non-planar configuration. So now you have the oxyhemoglobin, which is oxygen bound. It's in a relaxed state. It has more of a high affinity for oxygen, and it has a planar configuration. So binding capacity of oxygen in the lungs and capillaries. So the lungs, the pH level in the lungs is 7.4 and the torr is 100, which is partial pressure. It does not allow for salt bridges to form between the aspartate and the histidine, so which makes it more favorable when there are no salt bridges in between the aspartate and the histidine. Now in the capillaries, the pH is lower and the torr is lower, that which allows the side chains of the histidine beta 146 becomes protonated, which allows the salt bridge with the beta 994 forms, leading to a greater release of oxygen. So it's not good for oxygen binding. So it's going to release more oxygen, so it'll have a lower affinity for oxygen, as you see in the graph. So a decrease in pH causes a greater release of oxygen from the hemoglobin when it's a lower pH, which is, you see that the pH is 7.2, which in the capillaries is 7.2. So CO2 interacting with water can affect the pH levels of the capillaries. So when the carbon dioxide reacts with water, it's occurring inside of the red blood cells. CO2 reacts with the water to form carbonic acid. So you have the CO2 reacts with the water, which is catalyzed by the carbonic anhydrase, which it forms carbonic acid. Now, when it dissociates, it creates the bicarbonate and the hydrogen, resulting in a drop in pH in the inside of the cell. So that pretty much wraps up my presentation. I hope I was able to explain. I hope I didn't go too fast. I try to get in as much information as and be as thorough as possible. I hope this was helpful, and I look forward to watching everybody else's video. Thank you.